support this rule idea. So 3 of them. So the first one is about climatic variability or seasonality. So climatic variability or seasonality is greater at higher latitudes, right? So I already mentioned that the tropics, things are pretty steady, right? Temperature-wise and stuff, right? But when you get higher up latitudes, there's seasonality, there's summer and there's winter, right? So there's lots of variation. And so only organisms that have a broad range of tolerances, remember our tolerance curves we talked about broad versus narrow, right? Tolerances. So only animals that have a broad range of tolerances are going to be able to live at those more temperate and temperate regions, right? Or polar regions. So as a side effect of this broad tolerance range, then high latitude species can occupy more space. They can have broader ranges because they have a more broad tolerance, okay? So this hypothesis then, that explanation is going to predict that for terrestrial animals, that climatic tolerance should increase then from tropical to more polar areas, right? So again, it's reflected in the tolerance. So animals that have a wide tolerance can have a wider distribution, right? So, that can hold up for, we can see that in terrestrial animals. So it's been observed actually for amphibians. They've been tested and it shows this. But then it's interesting if you look at it from terms of, so that was terrestrial, but now think about it in terms of aquatic environments, talk about marine environments. So for marine fishes, where the greatest variability is not actually at the poles, right? The water at the poles is pretty consistent in its temperature, and the water at the equator is pretty consistent in its temperature. Where you have the greatest difference in water temperatures is going to be at more temperate regions. And so here's a graphic explaining that then for marine systems. So on the x-axis, we have latitude. On the y-axis, we have critical temperatures. So we have critical thermal max plotted here and critical thermal minimum plotted here. And now where you see the greatest difference is here at about 40 degrees latitude. So this is where then you would expect the fish species to have the broadest geographic range, because those are the fish that are most tolerant of a wide range of climatic conditions. Okay, so the way it works in aquatic systems, because of the properties of water, is a bit different, right? Because in the polar regions, it's cold there all the time. There's not much variation. At equatorial regions, it's warm there all the time. There's not a ton of variation. But where you see more variation is in the more temperate areas when you're thinking about aquatic systems, right? And that's where you see the greatest difference between CT max and CT min values. And so the fishes that are occupying those areas can have broader geographic ranges because they have a wider tolerance of temperatures. Okay, so again, thinking in mind the way things happen terrestrially is going to be different than the way things may happen in marine aquatic environments. Okay, so that's the first one. It has to do with climatic variability. It has to do with those ranges of tolerance. The second explanation has to do with glaciation and dispersal. So it's the suggestion here is that Rapoport's rule is the product of glaciation. And when the glaciers retreated, only those species that had really high dispersal capabilities were able to repopulate the northern areas as the glaciers retreated. So that means that, everybody got pushed down by the glaciers. But then when the glaciers receded, some species with greater dispersal capabilities could sort of track the retreat of the glacier. So you still have populations down here, but you have populations that are going more north because they have greater dispersal capabilities. So they've repopulated the northern areas. And so then they have this wider geographic range distribution. And so this idea, so there's, for every hypothesis, you can support it or refute it with evidence, right? So in this case, it seems to hold up pretty well for the northern hemisphere, where we actually had glaciation. But in the southern hemisphere, it doesn't quite hold up because we didn't have glaciation in the same way as we did in the north. So, there's some support for this idea in the northern hemisphere, but not so much support for it in the southern hemisphere because the glaciers did not have nearly the same extent as they did in the north. Okay. And then the third explanation for these patterns is this lack of competition in polar communities. And so this is based on the idea that there are fewer species, right? Overall, there's lower biodiversity, which we've already talked about, right? There's a lower biodiversity in these colder regions. So then there's less competition. Okay, so fewer species, less interspecific competition. So ranges can be bigger because there's less competition. Animals can spread out, right? So there's no support for this mechanism though, because we don't yet have a very simple way to measure competition. So it's hard to apply this on global patterns. So it's a hypothesis that is sort of at this point not been empirically tested with a great deal of science, right? Because it's hard to figure out how to quantify, to mechanistically quantify competition in a broad sense like that. So this hypothesis hasn't been effectively tested yet. But those are the three explanations for why we might see these patterns defined by Rapoport's rule. Okay. So, another question you might ask is there a relationship between this geographic range size that we've just talked about and the abundance? So, we're talking about how many individuals. So, for example, if a species is widespread, is it always going to be an abundant species? Do they always go together? So our widely distributed species always, high densities, high abundance. Or conversely, if a species is rare, so if you have a rare species or, like a threatened species or an endangered species, does it have a really small geographic range? Is there a relationship there? And so some data that's been collected by ecologists on plants and animals so far shows that there is a correlation between distribution and abundance, such that more widespread species are typically more abundant. And this pattern has been called Hanski's rule. So this more widespread species, so with broader geographic ranges, they tend to be species that are more abundant. So there's lots of them. And again, it's called Hanski's rule. So here's an example here from the Krebs ecology textbook. And this is the relationship between the distribution and abundance for 263 species of moths. So lots of data here, okay? So 263 species of moths. And what the pattern shows, so you have distribution and you have abundance. And so it shows that the more widespread moths are more abundant. You can see there's lots of variation around this line, but there is this general trend that the more abundant species are more widely distributed. So it seems to be support for this Hanski's rule in these moth data. Okay. And so again, it's a roughly positive linear relationship. But again, I will point out that there's tons of variation. Like this guy is, way out over here, and this guy is way down over here. But overall, there seems to be generally a linear positive relationship here in support of this idea of Hanski's rule. Okay. And so there's also three reasons why why abundance and distribution may be correlated. And so again, this is basically three hypotheses for why we might see this pattern. Okay, and the first one is the sampling model, which honestly is not really a hypothesis. It's more about a default explanation based on how we survey. So the sampling model is arguing that the observed relationship is simply an artifact of sampling. And it doesn't require then a biological explanation. It's just because of how we sample, right? So basically, rare species are more difficult to find, right? So rare species are hard to find. Common species are easier to find by definition of being rare and common. So if the appropriate biological studies are not done carefully, then you're going to automatically observe the predicted pattern, right? Because rare things are hard to find. So it's kind of a circular argument, right? And it's just about the sampling. So this, again, it's not really a biological explanation in that way. It's not really a hypothesis. It's more just talking about, well, it just has to do with an artifact of sampling. Okay, so it's difficult to evaluate that explanation because of this problem of counting things that are rare. It can't be the explanation for the pattern in. You could argue that this isn't the explanation for groups of animals like birds and butterflies and mammals, which have been actually really well studied, even the rare ones. Anyway, again, this first one is just kind of an excuse for why you might see the pattern rather than a biological explanation. Okay, the second one is more of a biological hypothesis. So it has to do with ecological specialization, which is called Brown's model. So this argues that species that can exploit a wide range of resources, so generalists, right? Generalists become both widespread and abundant relative to specialists. So, the idea being that the widespread generalist species are also going to have to use food and habitat resources that are also really abundant and widespread. But that's one idea, is that it has to do with this being a specialist versus a generalist. Okay, so that species, again, that can exploit a wide range of resources, that is, those that are generalists, can become more widespread and abundant relative to specialists. Okay, and then the third one is a sort of a metapopulation explanation. So this local population and dispersal model. So this suggests that a population is going to be in the format of a metapopulation. So we're going to have patches with demes, those subcomponents of a larger population, right? The demes are the locally breeding populations within a larger metapopulation. And so it suggests that in this system of a metapopulation type of system, that animals are going to move between those patches, right? And so since species differ in their dispersal capabilities, then some species are going to occupy more patches than others, right? And so if that's true, then we'd expect species that disperse more to be more common, okay, and widespread compared to less mobile species. So this has to do with a metapopulation sort of explanation and dispersal capabilities. So then species that can disperse more are going to be more common because they can go to more patches. And so they're going to be viewed as more common. So those are three ways to try to explain this pattern of more widespread species typically being more abundant. All right, the next one is Allen's rule, and I'm pretty sure you've learned about this one before, maybe in Principles of Ecology. And so now here we're considering the problem of heat loss by animals that live in cold places, right? You don't want to lose your heat if you're in a cold place, especially if you're an endotherm, right? So most heat is lost near the body's surface, right? So if there's less surface area then relative to volume, then less heat is going to be lost. So this has to do with conserving heat. And so it happens that just by physics, the geometric form in nature that has the least surface area relative to its volume is a sphere. A sphere, right? So this is a sphere is a shape that minimizes surface area to volume. And if you're trying not to lose heat, then you want to minimize your surface area to your volume, right? So this means that if you live in a cold place, you're going to find an advantage to being short and stocky, to being rather round in shape. So this is the case if we look at things like walruses and snowshoe hares, and I'll talk about some specific examples here in a second. So let's look at this one. So here we have two related species. We have the Blacktail Jackrabbit and we have the snowshoe hare that lives here in Sudbury. So the Blacktail Jackrabbit, this is a species that lives in really hot, arid conditions. It's a desert species. And if you look at him, he's got really long ears. He's got really long, skinny legs. He's got a longer, skinnier body. He's got a relatively long face, right? So he has a lot of surface area to volume. And that could be advantageous in a warm climate where he would want to lose some heat so he doesn't overheat. But then if you look at the snowshoe hare's body morphology, you can see that it's a much rounder body form. The ears are shorter, the face is rounder, the limbs are shorter, right? So this would be a more advantageous body form in a cold climate if you're trying to minimize the amount of heat that's lost across the body surface. Similarly, if we compare a red fox to an Arctic fox, a red fox has longer limbs, it has pointier ears, it has a much longer, pointier face compared to this more spherical shaped Arctic fox, right? It's got shorter, wider legs, a much shorter face, and so less surface area across which to lose that very expensive to generate body heat, right? When you're an endotherm. So in ecology, there's the rule that recognizes this principle of variation in body shape among geographic races of species, and it's called Allen's rule. And so what it states is that certain extremities of animals are relatively shorter in the colder parts of the species range than in the warmer parts. Okay, and again, this is based on the idea of heat conservation, right? So by extremities, as I've already pointed out, we're thinking about things like ears and noses and legs and tails, maybe, right? So those are the types of appendages we're talking about. Okay, so if you have shorter appendages, there's less surface area. Okay. So still talking about spheres and the advantage of being spherical. So it's also a matter of basic physics principles that a larger sphere has even less surface area per volume than a smaller sphere. So large spheres are going to lose heat more slowly compared to their size than a small sphere. So you might guess then an animal might be a big sphere in a cold environment, right? So being a big sphere is advantageous, but also then taking it a step further, being a big sphere would even be more advantageous in a cold environment. And so that's what Bergmann's rule says. Bergmann's rule asserts that geographic races of a species possessing smaller body size are found in warmer parts of the range, and races of larger body size in cooler parts of the range. So again, big spheres in cool places and smaller spheres in a little bit warmer places. So the rule predicts a pattern of body size variation with latitude, again, because we're talking about latitude as a surrogate for temperature, right? So we're looking at it as a latitude surrogate again, like we did at the start of our discussion today. And so in the course of the early 2000s, it was pretty interesting. There was a ton of research done on this idea, testing Bergmann's rule across many different groups of animals. And you can see that the first author on all these papers is Kyle Ashton. And so he was a young scientist at the time who was working on this as part of his PhD dissertation. So he wanted to tackle this idea of Bergmann's rule across all these different animal groups. And I'm not suggesting you have to read all these papers or anything like that, but just to show you that he did this really big comprehensive assessment and found, as you might expect, that there's some support for the rule and there's not always support for the rule, right? Rules don't always hold up. So, he's got this overarching thing that's about, are these rules being dismissed prematurely? But then he's got patterns in birds. And so it looks like birds follow Bergmann's rule. But if you look at amphibians, you might find something different. This is in a Canadian Journal of Zoology. And here's one about mammals. He questions whether or not it holds. And there is some support again. And then finally here in non-avian reptiles, turtles follow it, lizards and snakes reverse it. So it's a pretty neat assessment. I thought it was really interesting because I personally am interested in this idea of Bergmann's rule as well. And we'll come back to it a little bit later in the lecture when I talk about a test I did of Bergmann's rule with my research. Okay, the next one is Gloger's rule. And this rule says that dark pigments increase in races of animals living in warm and humid habitats. And so less pigmentation in the skin, basically with an increase in latitude. So that is, most animals tend to be lighter colored at higher latitudes. And among animal species supporting this includes humans. So this rule is supported by humans in that, generally speaking, dark-skinned human races tend to occur in more warmer environments, and light-skinned races tend to occur in more cooler climates. And so this is a map from the internet that shows that general pattern, that there's darker pigmented skin in the more warmer areas and paler pigmented skin in the cooler areas. There's some notable exceptions to this rule. For example, you think about ravens that live here in Sudbury and stay here all winter, but yet they're dark colored at this relatively cold latitude, and other mammals too, like martens and fishers and wolverines, which are also cold climate mammals that have dark fur color. And then there's, of course, polar bears which have this sort of ambiguous support, right? Because they have white fur, which supports the idea of Gloger's rule, but then they have black skin, which refutes the rule. And that's a whole separate sort of issue, and I'm not going to talk about it right now, something I may talk about a little bit more in winter ecology, but polar bears are kind of this interesting thing of their own accord. And so one of the explanations for in birds other than ravens, but some other birds that have dark colored feathers living in warm places, is that there is a relationship between the pigmentation and their ability to be broken down by bacteria, which is this first sub-bullet that's here. So one explanation for Gloger's rule in the case of birds is that there's an increased resistance in dark feathers to bacteria. And of course, bacteria love living in warm, humid environments. And so for birds living in warm, humid environments, they would find it advantageous to have dark pigmented feathers because the bacteria don't work as well in these dark pigments, right? So humid environments have higher bacterial loads, like humid, warm environments. The microbial growth loves that kind of situation. So dark feathers and even dark hair, if you talk about mammals, are more difficult to break down by bacteria. So there'd be some advantage to having dark pigments in that case. And also for mammals, there's a tendency for equatorial and tropical regions to have darker skin color, and then polar relatives to be lighter in color. And again, the hypothesis here is a protection against UV radiation. But there's absorption of certain amounts of UV radiation is necessary, though, for the production of vitamins like vitamin D, right? So there's some complications around that explanation as well. And again, as I mentioned, there seems to be a pattern in humans related to this, but there are also some exceptions to that rule. For example, Tibetan and Inuit peoples, which tend to have pretty dark skin, even though they live at higher altitudes and latitudes, higher elevations and latitudes. And this might be have to do with absorbing UV radiation and to complement the high vitamin D diets of people. But, so again, these are sort of general patterns, and there's going to be evidence to support and evidence to refute. Okay, what time it is? 10:45, we're good. Okay, so the next one to mention is the egg rule. And so this rule was proposed with special reference to birds in North America, in the Northern Hemisphere, I should say. And so this rule states that the average number of eggs in a clutch, so in one batch of eggs laid by songbirds and several other birds, increases as you move north in latitude. So larger clutch sizes in the north. Again, this is a discussion about Northern Hemisphere in this case. And so this probably evolved to balance the fact that in the north, where you have really short summers relative to more central latitudes, that birds might be able to only produce one successfully complete production of one clutch of eggs to fledgling, and therefore they would maximize the clutch size in that one clutch that they can produce. Because further south, where the active season is longer, birds can probably produce 2-3 clutches of eggs per year and get them to successful fledging. So again, in the south, the longer summer allows for multiple clutches per year. In the north, probably only time for one clutch per year. And so then birds will maximize that clutch size. Okay? That's the hypothesis proposed for birds. We see this hypothesis supported in other animals as well. So this is geographic variation in spotted turtles. This is some of my own research again. So we see this pattern, this egg rule, in freshwater turtles in North America, where we see a variation in clutch size with latitude. We see larger clutches in the north, but lower clutch frequency compared to the south. So go through this table here. So we have different variables here. In the first column, we have nesting season, the location of nest sites, which I'll talk a little bit more on the next slide. Body size. So this is plastron length, so lower shell length in millimeters. Clutch size, which is number of eggs in a single clutch. Clutch frequency is the number of clutches produced per year. And then we have total eggs produced per year when you account for clutch size and clutch frequency. Okay, so first we have South Carolina, which is a more southern population. These are data I collected during my PhD. And then we have Ontario, a more northern population. These are data I collected during my master's. And then we have here Pennsylvania, a more central population. And these are the data that were collected by Carl Ernst. And so these are data from the literature. So the first thing you can see is that in the south, nesting season is indeed a lot longer. It's about a 2-month period, whereas nesting season in Ontario is only about 2 weeks, so a much shorter window of time for turtles to lay their eggs. You can see across for body size is much bigger, and it's significantly bigger than in the central and southern populations. In fact, these two body sizes don't differ statistically. That's what the common A's mean. They're the same. The B is different, so that means that this value is different than these two values. That's a common way to use subscripts to explain your statistical analyses in a table. Here we have clutch size. Clutch size differs across the board. Smallest in the south, middle-sized in the middle, and largest in the north, supporting that idea of an egg rule. Clutch frequencies also supporting that idea of the egg rule. Clutch frequency is higher in the south. The turtles can lay, they often lay 2, and there's even a couple of cases of somebody laying 3 clutches of eggs in a single season. Pennsylvania, it's about 1 clutch per season, but at the Ontario site, females would actually skip reproduction some years. They just couldn't nest every year. So clutch frequency is less than 1 per year. But what I find is interesting, when you account for both clutch size and clutch frequency together at all these locations, the actual number of eggs produced per female per year is almost the same. It's about 3 eggs, maybe 4 eggs if you round up. So basically, each female produces 4 eggs per year, but those females are partitioning those egg resources differently. So it supports this idea of the egg rule being driven by this nesting season duration aspect. And sort of just to go with that, this is a comparison of the types of habitats in which females lay their eggs across spotted turtle populations. So again, the Georgian Bay, the northern site, the South Carolina site, and then here, for a central location, I have another one of our study sites that's along the shore of Lake Huron. So it's sort of a more southern location, but in Ontario. And so what's interesting is that the turtles in Georgian Bay, and all the turtle species do this here, they nest on rocks, which is not what you usually think of for turtle nesting. When you guys think of turtle nesting, I imagine you think about a sandy beach with a bunch of sea turtles coming up and nesting on that beach. You probably weren't thinking about turtles crawling up rock outcrops and laying their eggs on the rocks, but that's what they do. So here's a female spotted turtle here. She's sort of hiding under this lichen. Here's her cloaca with an egg popping out into this nest chamber that she has excavated from the little bit of soil that is right on the rock outcrop. And you contrast that with the female in South Carolina who's laying her nest in this rotten woody debris. This is, you can sort of see it here, but this is actually a rotten stump within the forest. So here, this is a fully exposed elevated rock outcrop. And here, this is a canopy closure swamp in a rotten piece of wood. And then at the Lake Huron site, they nest in the open floodplain. So there's some vegetation, but it's not a rock outcrop and it's not within the forest. So you have this variation across sites. And I think that that's actually really interesting and is probably driven by the thermal requirements for nesting, right? So again, in this northern area where you have a short growing season, using these great big rock outcrops, which probably have pretty amazing thermal inertia, that's a good place to put your eggs to make sure they're going to incubate and they're going to be hatching before winter comes. Whereas in South Carolina, the sort of selective pressure on females might be to pick places for their eggs that they're not going to hard boil and be overcooked, right? So they use the sort of damp canopied forest areas to produce their eggs. It's just sort of an interesting observation that goes with that egg rule stuff for these guys. Okay, so I mentioned that I wanted to talk about a more direct test of Bergmann's rule with some of the data that I've collected. So I'm going to discuss this paper that we published on Bergmann's rule about body size variation in spotted turtles. So body size variation, I mean, it's interesting from an evolutionary ecology perspective because there is this relationship between body size and reproductive output that we just saw, right? This egg rule. So if you're interested in evolutionary ecology questions, this is a really big deal, right? When you want to understand this. And of course, it has implications for conservation as well. Okay. So if you remember, Bergmann's rule says that geographic races of endothermic species are larger and cooler, so northern, than in warmer southern environments. So this idea, again, is about adapted heat conservation, that if you're a big sphere, you have a low surface area to volume ratio, so you're going to retain the heat. So this idea has been applied to ectotherms, which of course is a different scenario because they're not generating their own heat. But that's not to say that they don't want to retain the heat that they've acquired from the environment as well. And so here's the predicted pattern, right, that you would expect to see for Bergmann's rule, that with latitude moving from south to north, you expect to see this increase in body size, right? So I used the spotted turtle to test this idea. And so I want to talk a little bit about why turtles might be a good animal to use in general for this kind of a test. So they're good model organisms for this sort of thing because for tests of evolutionary ecology sort of theories like this, because it's really easy to permanently mark them. So that's one thing right away. So they have this hard shell. This upper shell is their carapace. You can see a little dent right here in this turtle's shell. That's just you, and you learned about this from Stephanie's video. and other ways that we've talked about marking turtles. You just use a triangular wood file and you can notch the edge of the shell, and every turtle has a different combination of these notches, and they're permanently marked. It doesn't harm them. And you know who it is, and the marks last for decades. And it's really inexpensive way to do it. You just buy a $5 file at Home Hardware and away you go. And again, everybody has a different combination of these notches, so you know who it is. So really easy to do. Turtles tend to occur in discrete populations in pretty large densities sometimes, so you can get a good sample size, right? You can get enough individuals. Another thing, if you're talking about reproductive output, that's super convenient that we do with turtles is you can X-ray them to get an estimate of clutch size. Because finding turtle nests in the wild is really, really hard and extremely labor intensive. And for this particular species, it's extra hard because they nest at night and they nest all night long and they nest individually. It's not like they all come up on a beach at the same time. There's one turtle nesting at one place and one turtle nesting way somewhere else. And again, it's the middle of the night. So it's hard to find individual nests. But if you can gather up all the females at the time when they should all be gravid, you can go to the vet and you can X-ray them and you can get a count of how many eggs they have, release the females, and off they go. So here's an X-ray here of a female who's gravid with 5 shelled eggs. You can see this is her radio transmitter on the back. So it's a really great way to get clutch size. And you might be thinking, wow, when X-rays are sort of dangerous and we wear lead capes when we do X-rays, especially pregnant female humans are not supposed to be near X-rays. So research was actually done to test whether or not there's an effect of this irradiation on females, on the eggs. And there's a published paper and it shows that there's actually no effect. The hatchability of eggs of X-rayed females is no different than the hatchability of eggs from un-X-rayed females. And in fact, the raw data show that hatchability was higher in the X-rayed females, but statistically speaking, there was no difference. So we know there's not an impact when we collect data this way. So it's a super convenient way to get a lot of reproductive information without, like I said, having to follow every single female all night long for in the South, for example, for 2 months. Okay. Also, they're large enough for attaching radio transmitters. So here's a glued on radio transmitter, and here's another style that's wired onto the edge of the shell. So that's a convenient way to find individual data information, as I've said before. And the other thing that's important for this body size comparison is that it's really easy to measure the body size of a turtle. They have this hard shell, and that hard shell doesn't change with short-term fluctuations in health, right? And turtles have fairly deterministic growth. That is, once they get to be adults, they don't grow very much anymore. So you can use calipers, put it on that shell, and get a really good measure of the body size of this animal, and repeatable. So not only is it easy to do, it's highly repeatable. You can, like I said, set calipers, which is a way to measure the length on there, and get really good measurements of body size. So that's why turtles are great for this kind of an assessment. Okay. The other thing I wanted to mention is that, the other studies that I talked about that measured, those Kyle Ashton studies that measured Bergmann's rule across different animals, when he did his research, he was doing groups of related animals. And so what I wanted to do was take a different approach and actually test it in a single species that's relatively wide ranging. So back to Cameron's question. So this is a geographic range, right? So this is what I'm talking about when I'm talking about geographic ranges, the distribution map for that species. So the spotted turtle is actually quite a wide-ranging species. It goes from 46 degrees north all the way to 28 degrees north. At least historically it did. Populations here in Central Florida are in trouble. But so we have this wide-ranging species. So this is actually interesting because this is a very rare animal. So although this is all colored in very solidly with red, there are not populations everywhere. There are not animals everywhere in this distribution. But this is just sort of the edges of where we know they occur. So it's a very rare species, yet it's widely distributed. So right away, here's an animal that refutes that Hanski's rule that I just talked about, that widely distributed species are common. I would argue that this is a widely distributed species that's rare. So that interesting sort of side test there. Okay, and the other thing I wanted to point out was the 2 study sites where I collected data. So the northern site here on Georgian Bay, and then the southern site here in South Carolina, just so you can see where those sites are. Okay, so I needed to measure body size of all these animals right across that range. So I have body size measurements that span all the way from that northern point down to pretty far south. And I didn't go collect live animals there. What I did was use a combination of live animal data that I collected. And also I traveled around to museums and measured the body size of adult turtles in museum collections, which was actually a really fun and interesting thing to do. So for those of you that have noted where Natalie is, one of your GTAs right now, she gets to sit in the Canadian Museum of Nature amongst all these different collections, and she's measuring wolf skulls for her master's work. And it's just a really cool experience to get behind the scenes in museums like that. So that's what I did. I have live animal data for 366 animals from my study site. So my Georgian Bay site, I visited a site in West Virginia, my PhD site, and then I visited another site in South Carolina. So I collected live animal data there. And then again, I drove around all these different museums, and I had 452 adult turtles measured from these museums. And this was really great because I was able to measure the body size of turtles from populations that are probably extirpated now. So these far south populations that don't exist in the wild anymore, there were collected specimens in these museum collections. Okay, so in terms of the results that were found, so this is a relationship between body size and latitude. So we've got latitude on the x-axis and carapace length, so that upper shell measured. And you can see that in general, it is following Bergmann's rule, right? We have this prediction of a positive linear relationship. It is a statistically significant relationship with 818 individual turtles in the analysis. But one thing you might note is that the R-squared value is actually quite crappy. So only 15% of the variation around this line is explained by the line, which is not a very powerful relationship. And I think that part of the reason that the regression line is actually significant in this case is just because of an enormous sample size. So we're detecting this trend because there's such a huge data set. And, I wanted to look at this a little more closely. So not only did I do latitude, which I said is a proxy for temperature, I also plotted this carapace length against minimum temperature and also against maximum temperature for all of these locations. And the pattern's exactly the same. So we see this sort of shallow positive relationship. But when I looked at the data, I'm like, it seems to me like there's so much mess around this line. And also, it seems like this northern population where I have tons of data from that one spot is really driving, like pushing up that line. So I took out that data set, and then I looked at it again just to see what would happen. I sort of explored the data. Sure enough, there's no relationship anymore, right? It's just a flat line. There's still 721 turtles in this analysis, but the R-squared is like abysmal, and the p-value is now not significant anymore. So there's no relationship here, right? So without that most northern population, which seemed to be driving the whole thing, indeed, it was driving the whole thing. So this is still 16 degrees of latitude. It's still a pretty wide spread, but now there's no relationship. So Bergmann's rule doesn't seem to be there. But I wanted to dive in still doing this sort of exploration of data. And if you're doing a fourth year thesis, I highly encourage you to do this kind of exploration with your data and look at patterns that might be there. So the next thing I did was take the live and museum data, but I took out that northern population again. And then I did a different kind of analysis. I didn't do a regression. I did something called a cubic spline. And you don't need to know what that is, just that I tried another way to explore the data. And it gave this pattern where there's actually smaller turtles at this more sort of central latitude. So then I used just the live animal data. So here you could argue I have, bigger sample sizes at each different latitude. So this is all the live animal data. So now that northern population is still there. And I applied a fit of a second order polynomial, again, just exploring the data, but it shows a similar thing that the smallest turtles are actually at about 39 degrees latitude. So not at the most southern place, right? Bergmann's rule says that the smallest turtles should be down here at the south. And that in both of these exploratory analyses, that's not the case. The smallest turtles are actually in the middle part of the range. So these findings bring up two questions to me, like, what's going on? Always science brings up more questions, right? So here's a case of that. So, why are those, for two questions that came up for me. So first one, why are the northern turtles so big? Like, why would they be so much bigger than everybody else that they're actually driving that Bergmann's rule relationship in the first set of analyses? So there's three hypotheses that I have for why that might be happening. And it's related to some of the stuff we've already talked about. And one thing to note is that these three explanations are not mutually exclusive. So I told you in an earlier lecture in the course about the power of strong inference and having mutually exclusive hypotheses. I will say right up front that these are not, so it would be hard to tease these apart, but I think they're still worth discussing as possible explanations. So the first one has to do with seasonality, and we've talked about this as an explanation for other patterns, right? So I think it might have to do with this unpredictability and seasonality in the northern landscape. So larger body size is favored in seasonal environments where food might be temporarily unavailable because a larger individual can survive for a longer period of time during food shortages, right? There's more space, there's more fat stores, there's a bigger body size to weather those storms of shortages of food. So larger individuals can survive longer without access to food than smaller individuals can. So, more variation in body size maybe is explained by annual temperature range rather than by temperature per se. So it's the variation in temperature which is that seasonality piece. And in fact, that type of pattern has been found in, for example, house sparrows and antlions as other animals that seem to have that pattern. So again, seasonality, it's temperature variation that might be more important than, average temperature per se. And again, this idea that larger individuals can survive food shortages better than smaller individuals. So maybe that's what's going on in the north. Second one is that maybe it's related because they're not mutually exclusive, but it might be that so that if they're big, then they can hibernate and reproduce. So this is the idea that bigger body size means, again, more space for energy storage to survive the long hibernation period is like 6 months long, but still have energy left over in the spring when they emerge to put right into egg development to get ready to reproduce. Because again, it's a very short window of time for reproduction in the north. So the sooner you can ramp up your body to get your eggs ready and into the environment, the better chance they have of finishing incubation before winter comes again, before fall temperatures come and cool things off. So this is an idea that's called capital versus income breeders. So this would suggest that turtles are capital breeders. That is, they're using the energy from before to invest in reproduction now versus income breeders, which would be those that eat right now and put that right into their eggs, right? So there's not enough time for northern turtles to be income breeders. They have to be capital breeders. So if you're big, you can do it all. You can hibernate and reproduce based on the capital that you gained the previous year, that capital being energy. And then related to that again is this idea of maximizing clutch size, right? So we already saw from that table that I showed comparing clutch size across populations of spotted turtles that bigger females have bigger clutch sizes. If you think about a turtle, unlike a mammal, when a turtle is gravid, the number of eggs she can carry is totally dictated by her shell because that shell cannot expand. Unlike a mammal, right? When a mammal is pregnant, the female's body can expand out to accommodate the embryos developing inside of her. But for a turtle, that's not the case. So the only way a turtle can have a bigger clutch size is to have a bigger body. There's this very tight relationship, right? Because the shell can't expand when she's gravid. So this is the idea that if they can only reproduce once per year at most, because the summer's short, then there'd be an advantage to being as big as possible, so you can put as many eggs in that one basket as possible, right? So Ontario data show this strong relationship between body size and clutch size, and that relationship is not as strong in more southern populations. So there's an advantage then to delaying maturity until a bigger body size because you can produce more eggs. Okay, the second question that comes up is about why those small turtles are in the central part of the range instead of at the south where Bergmann's rules suggest that they should be. So why are the smallest turtles in that central part of the range? And here, I think that it has to do with female reproductive cycles and a change in allocation of energy. So we talked before also about energy budgets, right? Things that go to growth versus things that go to reproduction. So this dip in body size is reflecting a change in the energy allocation to growth and reproduction. So it's the latitude where clutch frequency changes. Clutch frequency changes from one or less per year to multiple per year. So at points north of that, clutch frequency is reduced, and points south of that, clutch frequency is higher, right? So it reflects this trade-off. It indicates this transition zone where clutch frequency changes to less than 1 above that point to greater than 1 below that point. And so basically the turtles then above this 39 degrees north, which seems to be this transition zone, they produce a maximum of 1 clutch per year. because they are instead putting some energy into growth to achieve that bigger body size to get through that winter and survive those food shortages. In the south, why the turtles start to get bigger again at the most southern places is because now there is a long growing season. So turtles can both reproduce and eat a bunch and grow a bunch because it's such happy living down there. So they can do both. They can both increase their clutches and body size. So body sizes is coming up again at that most southern location. So it has to do with sort of the productivity that's there. And this pattern in turtles is also reflected in other animals. So it resembles what has been called a sawtooth pattern that's in some invertebrates, terrestrial invertebrates. So it's an analogous situation where we have a switch from univoltine at more northern locations to bivoltine at more southern locations. So univoltine is 1 life cycle per year, bivoltine is 2, and then maybe even more, but bivoltine is 2 life cycles per year. So it's an analogous situation to clutch frequency. In this case, it's life cycles that can happen. So it's kind of interesting that might be what's happening at that central part. And of course, a way to actually test that is to go to populations at that range, at that 39 degrees north, and test it and see what the growth rates are and see what the clutch frequencies are and figure that out. I haven't done it yet. That would be going to North Carolina and testing what spotted turtles are doing there. Okay. So my former graduate student, Billy Greaves, who worked on wood turtles, he looked at body size variation among populations, and he found a similar idea. He found it's not linear. So he found again that wood turtles at the most southern part of the range are this size, but then the smallest ones again are at the central part of this species range, and the biggest ones are at the northern part of the range. So like the spotted turtles, he found this sort of checkmark shape instead of the linear pattern that would be predicted by Bergmann's rule. So it's not linear, so it doesn't fit it. It's, again, this sort of U-shaped pattern. And this is comparing it now to the clutch size. So it's totally reflected in that clutch size. Again, supporting that idea that body size and clutch size are so highly tied to each other for these freshwater turtles. Bigger females can produce bigger clutches of eggs, right? And then those central females have medium-sized clutches of eggs, and southern females, again, starts to get bigger because their body size is a bit bigger. So this is interesting to me because now this refutes that egg rule that we just talked about, right? The egg rule says that this should be a linear relationship as well. But I think, again, it's being driven by this body size pattern. Okay. All right. So a bit of a summary then of these ecological rules. Again, this idea that gradients in species richness are related to temperature. So then the total number of species generally increases from temperate to equatorial regions, right? That has to do with temperature. And the total number of species decreases with increasing elevation, right? So this says altitude, but it should say elevation to be a little bit more accurate. So we have this gradients again, right? So in warm places, which warm places can be equatorial or at the base of a mountain, you have higher species richness. And in colder places, that is higher latitudes or higher elevations up the mountain, you have lower species richness. Okay. All right. And then we talked about Damuth's rule of energetic equivalence, right? This is the idea that the amount of energy consumed by a given species is independent of the body size. And that's because of those exponents canceling each other out in their relationships. Okay. Rapoport's rule, this was the idea that geographic range sizes decrease as one moves from polar to equatorial regions, so that we have range sizes are smaller in the tropics and range sizes are bigger in more temperate and polar regions. So we have this pattern. And then Hanski's rule, this was the idea that more widespread species are typically more abundant, more common species. And then Allen's rule, which talks about reduced extremities in cold places, right? So certain extremities are relatively shorter in the cooler parts of the species range than in warmer parts. So this is that idea that it's better to be a sphere in cold places to minimize that surface area to volume relationship, to minimize heat loss. And so related to that is also Bergmann's rule, which says that a bigger sphere is even better than a small sphere when you're living in cold places, right? So geographic races of a species have smaller body sizes. Those are found in the warmer parts of the range, and races of larger bodied species are found in cooler parts of the range. So again, it's better to be a big sphere in a cold place. And then we talked in detail about how spotted turtles sort of don't fit that, kind of, depending on how you look at the data. And then Gloger's rule, this was the idea that dark pigments in the skin are more prevalent in races of animals that live in warm places and humid places. And then finally, the egg rule, this idea that the average number of eggs in a clutch increases as you go more north. So there's a relationship between clutch size and latitude. But again, this doesn't seem to hold up for everything. It was first proposed in birds. There's some support, but it seems, at least in the freshwater turtles, that it doesn't actually fit very well. And this picture is here just because it's a really awesome, gigantic toad. This is a picture of an American toad that I found out behind my house this past summer. This is my daughter's hand. And so she's got a pretty big hand. You can see the massive size of this female toad. And I have never, ever seen a toad that big. So I just thought I would share it with you. I was happy to find her living in Sudbury. Is that a Bergmann's rule thing? If I went south, would the toad be small? Okay. So I'm going to stop sharing my screen, and we are at 11:14. So we can talk a little bit more about the exams. Oh my God, there's 13 chats. Holy, sorry. Is there anything I need to know about in the chat? Let's see. Were you able to see your individual marks on things? Can somebody just maybe tell me out loud? I don't know if I can know these chats. Yes, Noah says yes. Okay, so good. I was finally able to do that on D2L, so that's good to know. Found a female American toad that might be bigger than the one in, what? Carol Ann, you found a bigger one than that? Are you sure? Honestly, I've been catching toads since I was a little tiny kid, and that was the biggest toad I've ever seen.
It was big. It took 2 hands to pick it up.
And it was an American toad. Where was it, Carol Ann?
I did my project in Hearst, so it was back home.
So way far north.
Yeah.
Oh, I would love to. Oh, man, that's so cool.
I can send you a picture if you want.
Yeah, please, with the scale of your hand in it. I want to show it to my daughter, too, because she's pretty excited that there's a picture of her toad in hand there in our presentation today. Will we ever do a correction of the midterm? Yeah, so it's 11:16. What I can do right now is at least tell you where I think the most important corrections are that you need to think about. So, the true false questions, they're pretty straightforward. If you got that one wrong, you know exactly what the proper answer is. And so those were largely based on the papers that I discussed. So, you can look at the slides related to those papers and figure that out. The definitions also, so for the most part, they were actually pretty well done. They were 2-point questions. So I was looking for 2 items basically for all of them. And they didn't have to be this, there could be 2 or 3 or even 4 points for some of them that would be valued at 2. But they also should be fairly straightforward. There was one that a lot of people got wrong, and that was the ecological energy budget one. And people talked about the biosphere, and it seemed a pretty consistent answer. So I wonder if it's something you learned about in another class. But ecological energy budget, that's the pie that I talked about, where the animal has finite energy and it has to divide that energy up among the slices of the pie, the 2 big slices being basic maintenance, right, and production. And then within maintenance, there's basic maintenance and digestion, et cetera, activity. And then in production, right, that's the part about reproduction, growth, and storage. So that's what that was about. Let's see. The question that I was really hoping for more on was the one about describing an ecological experiment that tells me what a hypothesis is, what a prediction is, or a research question at least, and that what sample size is, and that what replication is, and that what the problem is when you don't have a large enough sample size. So the answer there is random error, right? That is when your sample doesn't represent the population because your sample is too small. And you fix that by increasing your sample size. A lot of people said when the sample size is too small, the problem you have is a bias. That's wrong. The bias is what happens when you have systematic error. That is, there's some wrong way about the way you're collecting data that presents a bias. It has nothing to do with sample size. Okay, so that's and I gave 2 examples in class exactly related to this. We talked about clutch size in fishes in lakes of different depths. And we talked about urban raccoon body size versus wild space, natural habitat body size. So I gave two very explicit examples about this. So in some cases, people proposed a research question or idea that in fact would be impossible to test in a way that you could answer this exam question. So that one I really want you guys to think about more. That one needs some rethinking. And it's a really important piece of what I'm trying to teach you in this course. So I really want you to think about it again. So what I'm telling you is I'm going to ask it again on the final exam. So I want you to understand that. Okay. So I've told you right away already what I'm going to re-ask on the final. Okay. The questions that had to do with math, like the one about the metapopulation of voles and the one about the Ministry of Natural Resources managing Algonquin wolves, those two questions were based entirely on example calculations that we did in lecture and also on the study questions that I posted on D2L. So if you did those study questions, you would have been able to do those questions because they were basically the same with just different numbers. So doing the study questions was essential to answering those exam questions. And so it was really obvious when people had done the study questions for those. The K/2 one, the graphic that didn't show up, I'm super frustrated that didn't work. And I'm guessing it had to do with lockdown, which when I set up the exam on D2L, there was no indication from D2L that would be a problem. So I'm really frustrated with that. But that said, you didn't need to see the graphic to answer the question. But as I said at the start of lecture, I can understand that would totally freak you out if you couldn't open the link and you needed to try to answer a question about it, even if you didn't need to see the graph. So that's why I gave that question was why I gave an extra 5% to everybody, recognizing that. And then also the life table question, because I recognized that it would be hard to type some of these formulas. So for the most part, the life table question was actually done fairly well because I told you guys explicitly there would be a life table on the exam. So I was hoping that you would expect I would exactly ask these types of questions about a life table and to fill in some of those calculations of the life table. I'm not going to ask that question again, but I'm hoping that it's that you did learn something about life tables. But again, I'm not going to ask that again on the final exam. And what I will say also is that if I do ask more calculations to be done on the final exam, I'll give you the equations just to try and ease that stress a little bit. Lots of biologists still look up stuff all the time, right? So I don't think it's necessarily a valuable thing for you to have to memorize every single one of these equations. And so, and I sort of wish I would have done that now for this midterm, but I can say to you guys, honestly, this is a learning curve for me too. I've never had to teach this course on D2L before. And I can tell you from my perspective, it's really hard. It's really, really hard to teach you guys in this context. So I'm assuming it's also not very easy for you to learn in this context. So it's a learning process for me on how to deliver an exam in D2L. I wish we didn't have to do it this way. I'm sure you wish it as well. Thank you, Cameron. I'm trying to, right? And I also wanted to try to get the exams marked as fast as possible because I knew that there was a drop date pending, but I think I missed it because I think the drop date was Monday, right? But anyhow, we'll work through this all together. But I think one of the key things is you guys need to be here at the lectures. You need to be paying attention to what we're talking about at the lectures. And I know that that's hard when we're doing it online. But let's really try to make sure that the stuff is, sinking in. I mean, my point is that you guys learn something. My point isn't that you have to get, a 95% on the exam. My point is that I want you guys to learn stuff about animal ecology because that's why we're here. So we're past time, but if you guys, a lot of you are still here. Does anybody have any specific questions about a question on the exam? Like I tried to cover a bunch of stuff related to it, but is there anything specific that anybody wants to ask about on the exam?
Would the actual exam be the same format?
Yeah, so probably there will probably be short answer questions and medium answer and long answer questions. One of the things I generally like to do is to have choice And that's hard to do on D2L without having a bunch of confusion, but I'm going to try to figure out a way to do that. So what I'll say related to that is that you really look closely at what the question is asking. So I did this at the end of the year last year when I had to do the histology exam. All of a sudden on D2L, I wanted an essay on there, and I did do choice, but I had to very clearly write it. Like, so it's one question, but there's a big bolded OR in the middle to say, you can do this part or this part and only answer one of them. So I'm going to try to figure out a way to do that for the final. So you'll just have to really read carefully. So there's a few chats coming in. Could you go over describing the experiment question again? In a second, when we have more time. Will we have more time for the final? Yes. I wasn't really sure how to answer the short answer questions. I did some as a list and some in essay. And that, bulleted lists are fine. I know I could tell a lot of you were typing really quickly because the spelling was really funny. And that's okay. I'm not totally worried about that when we're trying to type quickly. So either way, Benjamin was totally fine. Okay, so back to, Jackson asked about describing experiment. Well, I'm not going to give you the answer all over again. There are 2 examples that are in the lecture, but I really what I'm wanting in that question is I want to know if you understand what a hypothesis is, what an ecological study is, what a hypothesis is, and how many you need to have as your sample size, how many you need to have as your replicates. So I want you to really tie together. If you describe to me an ecological experiment that you're doing, make it really clear in that question, what is the sample size? Like, what are you sampling? What's the response you're measuring? Is it body size? Well, then tell me you're measuring body size for 30 individuals in each of your replicated sites being 5 urban habitats and 5 natural habitats. Okay. Like, I want it to be very explicitly described as an ecological experiment. So I hope that answers your question there, Jackson. But also check the lecture notes, because I give 2 examples there, the raccoon example and the fish clutch size example. Will the exam be through D2L with lockdown again? I don't know yet. Lockdown's a pain in the butt. I'm just not sure if I'm going to do lockdown again. It's an effort to try to get to minimize cheating, but I realize that cheating is really easy to do anyhow. So I'm trying not to stress about that. I mean, my emphasis again is I want you guys to learn stuff. Like, that's the most important thing to me. So I'm hoping you don't cheat, but I'm not going to stress about you cheating, because you know what? There's nothing I can do about it. If you cheat, you cheat. And I really can't do anything about it. And I think it's super invasive to have you write an exam with the lockdown camera thing. Like, I just think that's kind of creepy. But if I was with you in person, I'd be watching where your eyes are roaming and what you're doing. So it's just, this is what we've got. So to answer your question, Jenna, I'm not sure yet if I'll use lockdown again. But if I do, I want to make sure that you guys know in advance, so that you have to have the right device to write on. Lockdown cannot work on a Google Chromebook, or a Chromebook, so you wouldn't be able to use a Chromebook for the final. I found that I did not have enough time to check my work on the exam, as I found it quite long. Yeah, and so that was also a challenge, and so that's also why I added 5% to everybody's mark, and I can tell you honestly, in the 16 years I've been teaching, I have never done that before, because I felt like the exam tested fairly in the content, but because of this online situation we're in, I gave everybody 5%. And as I said, I've never done that before. I usually stick to my guns pretty hard about that kind of thing, but this is such an unusual situation that I gave it. So I recognize the time challenge. And again, part of that was I was trying to make sure people weren't cheating as well. But again, I'm gonna have to rethink that. But I will keep you posted for sure. Multiple choice, no. Multiple choices, no. I don't think so. I do that for histology because there's 125 students in the class, and I can't mark 125 exams. In fact, marking these 68 exams for you guys was incredibly time consuming. So I'm not going to do multiple choice, because I don't think they actually are super effective, especially in a course like this, where my point is to figure out if you've synthesized information and understood it. That's different than memorizing. So, which is why I made the comment too, that I think if there's any more mathematical stuff, I'll give you the equations, because it's not valuable to memorize equations. it's valuable to understand how to apply them and answer a biological question. So I do regret that I did not give you the equations, like, for example, for calculating exponential growth, because that's not the point of what I'm trying to have you learn. Okay. We are now getting pretty late on time. There's still a few of you here. Is there anything else that anybody wants to ask? I'm really glad that you guys can finally access your exams.
Thank you so much for all the feedback.
You're welcome, Lucy. Okay.
Thanks, Jackie. Have a good day.
You too, everybody.
Thank you very much. Appreciate it. Have yourself a lovely day.
Enjoy the sunshine, warm weather.